I'm going to start the next tutorial on how to store optical physiology data, such as 1-photon or 2-photon data, to NWB using PyNWB, and then Ben will cover the doing the same using MatNWB. You can get to this tutorial by going to the PyNWB website, the Read the Docs page, going to Tutorials. This will be the second tutorial under domain-specific tutorials. Okay, so in this tutorial, we're going to cover how to create an imaging plane and add it to the NWB file, how to add our acquired 2-photon images or 1-photon images, how to specify any motion correction that we are applying to our raw data, how to add image segmentation, and finally, the computed fluorescence or DF/F response traces for each ROI. Okay, so we'll start with a bunch of imports. Most of these imports are from PyNWB.offiz or PyNWB.image. And just like the last two tutorials, we will create an NWB file object by calling NWB file, passing in keyword arguments as such here. All right, so the first thing that we'll want to do is to create an imaging plane object. And this holds information about the area and the method used to collect the optical imaging data. This will require the creation of a device object first that represents the microscope, and an optical channel object that represents your optical channel. So first we'll call NWB file.createDevice and pass a name, description, and manufacturer for your microscope. We'll just take some example text here. And then we'll create an optical channel object with a name, a description, and the emission lambda of our channel. So then we will create an imaging plane by calling NWB file.createImagingPlane and passing a set of keyword arguments such as the name, the optical channel that we just created above, the rate in hertz, so 30 hertz, a description, the device that we created above as well, the excitation lambda, the indicator like GFP, where in the brain this was, and then additional parameters specifying the grid spacing and where is the origin of the imaging plane. Once we have this imaging plane object with all of these metadata describing the plane of imaging, we can create our 1-photon series object as the first example. So we'll just call the constructor for 1-photon series and pass in a name, a description, a data array. Again, in NWB, for all data arrays, the first dimension is always time. So in this case, we have 1,000 frames of 100 by 100 images. We pass in the imaging plane that was used, which is what we just created up here. And we'll say that the sampling rate is 1 hertz. The starting time is not listed here, but the default starting time is 0, which means that the first frame would start at the session start time that is described in the NWB file constructor here. And then we can customize those or specify explicit timestamps for each frame of the 1-photon series. Yeah, so once we've created this 1-photon series object, it's not part of the NWB file yet. We have because these are raw acquired data, we're going to add this object to the NWB file as in the acquisition group using NWB file.add_acquisition and passing in the object. 2-photon series data store 2-photon imaging data, as you might expect. And it behaves very similar to the 1-photon series object. when we're going to create it in basically the exact same way and add it to an NWB file object in the same way. We can also store the results of motion correction using a motion correction object, which we'll see down here. And to do that, we have to create a corrected image stack object. And this takes in a corrected image series, as well as the original image series, or the 1 photon or 2 photon series. And this motion correction works, or is compatible for describing affine transformations or translations, so XY translations in this case. And we have some separate types either in the works or in extensions to cover other types of motion correction. So here we will create an image series that represents the corrected image series. This one has to be named corrected because of how it's being added to this object. And then we'll pass in a description, a data array with your corrected image stack, and additional parameters here. And then we can also specify a time series that is the XY translation. So X is the first the first row and Y is the second row of this data. And one XY value for each frame in your image stack. And we'll pass these into the corrected image stack constructor and create a motion correction object that takes as inputs one or more of these corrected image stacks. So this motion correction.
Yep. Oh, there's just a quick question. The data in this case, it's just a placeholder, correct? That you would substitute with your actual data for these XY translation and corrected.
Exactly. Yeah. Sorry. This is these are placeholder data. Yeah. If you're working with your own data, make sure to replace these with your actual data. Okay. So this motion correction object and all of its child objects are not yet part of the NWB file. Because this is the results of processing the raw data, we're going to put this in the processing group of the NWB file. And to do that, we'll first create a processing module named OPHYS for optical physiology data and add this motion correction data to the file. So just like in the previous tutorials, you can call NWB file.create processing module, passing in a name, OPHYS, a description of the module, and then we're going to add our motion correction object to this module. This step is optional. If you have motion correction data that you want to add, you can do it this way. Sometimes your processing tool, if it can export to NWB, it will automatically have this as well, or do this on its own. Next, we're going to cover how to store the detected regions of interest in your 2 photon series data. And we will use the plane segmentation class. And plane segmentation is a subtype or subclass of dynamic table, just like the electrodes table, the units table, and the trials table. This is basically a table of ROIs. And each row represents a single region of interest. There are 2 primary columns, or actually 3 columns of a plane segmentation, and they are image mask, pixel mask, and if you're working with volumetric data, you can also have a voxel mask. And you're required to specify one of these that specifies the extent of the ROI in your image. You also have to specify the imaging plane, which we created earlier. Okay. And then I'll come back to the image segmentation object, or actually we'll start there. So this plane segmentation table will hold a bunch of ROIs, and you may run different segmentation algorithms on your data, and as a result, have different ROI segmentations of your raw data. And so you can create multiple plane segmentations, one for each algorithm or parameter setting. And you can store all of these different plane segmentation tables within another object called image segmentation. And image segmentation just holds one or more plane segmentation objects, just like in the behavior data, a position class or object holds multiple spatial series, or the LFP holds multiple electrical series. Here, the image segmentation holds multiple plane segmentations. So to get started with this, we'll first create an image segmentation object, and it will be empty, just like so. And we'll create a plane segmentation from that image segmentation, or within that image segmentation. And we'll pass in keyword argument parameters such as name, description, the imaging plane, and any reference images that holding the raw data. And this is optional. If you don't have the raw data, or you don't want to share the raw data, you can leave this out. We previously created a processing module for optical physiology called OPHYS, and so that is our OPHYS module. And so we're going to add our image segmentation object to our OPHYS module, and that has our plane segmentation, which is currently empty. Next, we'll add the individual regions of interest. So we can describe our regions of interest using an image mask or a pixel mask. The difference is that an image mask is an array that is the same size as a single frame of your raw data, your 1 photon series or 2 photon series, that indicates the mask weights at each pixel of the image. So, for example, if your image is 100 by 100, your mask would also be a 100 by 100 with values that range from 0 to 1. And so here we're going to create just 30 random ROIs. The image has shape 100 by 100. Sorry, the mask has shape 100 by 100, and we're just going to create a random 5 by 5 ROI in it. And we'll create the ROI by calling plane segmentation.add ROI and passing in our 100 by 100 mask. Alternatively, we can define ROIs using a pixel mask. This is a more sparse representation of the mask. This is just an array of triplets, x, y, and weight, where weight is a non-zero value. And then anything that is not defined, any pixel coordinates that don't have a triplet, a weight, will have a weight of 0. So in this case, we're going to let's just create a new plane segmentation here that describe where we're going to use pixel masks, and we'll loop through 30 ROIs. For each pixel mask, we will pick a random starting point and add the individual pixels for that mask, and give them all a weight of 1. And so in this example, we'll have a 4 by 3 region of pixels. So this pixel mask will have 12 triplets at the end. And then we're going to use the add ROI method, just like above, this time passing in pixel mask equals our pixel mask. If we have volumetric imaging data, we can use 3D imaging masks in much the same way, using image masks, but they are 3-dimensional instead of 2-dimensional. Or you can have a voxel mask, where it's instead of a triplet of XY weights, you have a what's the word? A quadlet of XYZ weight. All right, so then we will have our 30 ROIs in our plane segmentation object. And we can view those ROIs using the toDataFrame method, because this is a dynamic table just like the other tables. Finally, now we have regions of interest, we have our raw data, we can store the fluorescence data for each of these regions of interest. And these are stored in an ROI response series class. And this is a subclass of time series. So as a reminder, time series, it stores data, name, description, data, and either a rate and starting time or timestamps. The ROI response series specifies that the data must be time by ROI, and that we have a new array that is our ROIs, which references rows of a particular plane segmentation table. So if you had 20 ROIs in your plane segmentation table, you could reference all of those rows, and your ROI response series would show the fluorescence for 20 ROIs, or you could have an ROI response series that has a subset of those ROIs. So to create this dynamic table region reference that references rows of our plane segmentation table, we would call plane segmentation, access the table here, and call create ROI table region, and pass in a list of the indices of the rows that you want to reference. So in this case, we're just going to reference the first 2 ROIs of our plane segmentation table. But this could be all of them if you would like. And then we will create an ROI response series object that holds our fluorescence data. And using the constructor and passing in name, description, our data, and this is a placeholder here, but let's say we had 50 frames and 2 ROIs. Pass in our ROIs table region reference, which we created up here, which says that we're just referring to the first 2 ROIs. Pass in a unit of measurement and the sampling rates. Okay, so to help downstream data analysis and visualization tools know that this ROI response series is fluorescence data as opposed to, let's say, DF over F data, then we'll store this ROI response series object inside of a fluorescence container object, much like we stored our spatial series in a position object earlier. And so to do that, we create a fluorescence object and we pass in our ROI response series. And this fluorescence object is currently floating. It's not part of the file yet. To add it to the file, we will add it to our processing module for OFIS. If you want to store DF over F data instead of raw fluorescence data, you can put the ROI response series object in a DF over F object instead of a fluorescence object, and make sure that the data is actually in units of DF over F. Okay, finally, we will write the file in the same way as in the previous tutorials. We'll call NWB HDF5IO, pass a file name, pass a mode, W for write, and call io.write on our NWB file. To read data, we'll use the same class, passing in a mode of R, and calling io.read to get our read NWB file. And then we can access the individual elements that we added earlier by looking at the acquisition group and passing in the name of the 2 photon series, which is by default or by convention, the name of the object, 2 photon series. Our fluorescence data are stored in a nested structure under the OFIS processing module. So we'll go to read NWB file.processing, pass the name OFIS, and then it's in a group, the fluorescence grouping object called fluorescence, and then it's in the ROI response series object within that. Now, just like in the other time series data, these data arrays can be very big, and they are not read passively from the file. So when you print this out, you won't be reading all of your fluorescence data or your raw data into memory. If you want to do that, you can call the .data accessor on the object, and then and then slice all of the values using the colon here to get all the values into memory, or to store it in a variable. Often these data are big, so you might want to index or slice this data array and just get a subset of it. And you can do that using standard NumPy array slicing syntax. So you can, in this case, we will get the first the first 10 first 10 frames in the first 0, 1, 2, 3 ROIs. We'll get the fluorescence data for the first 10 frames in the first three ROIs of this example ROI response series. And that's it. So we covered specifying an imaging plane with the metadata about the actual imaging, including the microscope and the optical channels. We went over how to add your raw 1 photon or 2 photon data to the NWB file. We went over how to create an OPHYS processing module and create an image segmentation group that holds plane segmentation tables, which hold your regions of interest. And we covered how to store the time series that holds the fluorescence or DF over F data from each of those ROIs. Next, I'll pass it to Ben to cover much the same material using MATLAB.
All right, as before, I will go over the MATLAB side for optical physiology, which is really mostly just calcium imaging, or usually calcium imaging, and I'll try to emphasize the differences between Python and MATLAB. So this will be loading in your acquired 2 photon or 1 photon data, inputting your image segmentation results and your associated fluorescence or DF over F traces. So first things first, create the NWB file object, just same as the last 2 tutorials. Okay, so now we get into optical physiology. So first we want to add our 2 photon data. So in order to add 2 photon series, that refers to an imaging plane. So you need to create this imaging plane. This imaging plane contains an optical channel or one or more optical channels and a device. So first thing you need to do is create this optical channel and device, and then this imaging plane. Okay, so let's go down here. All right, so here we go. Optical channel, we'll just give it some filler values, description and emission lambda. Device, we're just gonna give it no values. Okay, and so then we set device inside of nwb.general devices. And we create the imaging plane with the name imaging plane using types.core.imaging plane. This optical channel gets set here. Notice this is a bit tricky. The optical channel is not a link because it is actually contained within the imaging plane, whereas the device is a link. It exists outside and is linked to within the imaging plane. Okay, so we have types.core.imaging plane, the optical channel, description, device, excitation lambda, which is in nanometers, an imaging rate. Now you're going to separately define the timing of the recording. So this imaging rate can be kind of approximate, or like your nominal imaging rate, because sometimes there's a bit of jitter. The indicator used and the location, which should be an area of the brain. So once we create this imaging plane object, then we can do nwb.general optophysiology.set imaging plane name, which we have here, to this imaging plane object we've created. All right, so now we have all the metadata that we need to actually add this two photon series. And it's similar to spatial series and to electrical series in that it inherits from time series and adds a bit of metadata. In this case, it's just linking to the specific imaging plane where it was recorded. So, and that linking is done again with a soft link, types.untyped.soft link, and then you give it the imaging plane object. And then you have the choice of doing it with starting time and rate or with timestamps, just like every other time series. Here I'm showing with starting time and rate. Again, rate is starting time, starting underscore time underscore rate. The data here, remember, time is on the last axis, which is different from Python. And data underscore units, we'll say lumens. And then once we've created this object, we can add it to the NWB file under acquisition with nwb.acquisition.set. I'm setting it just to photon series, and then I'm giving it that variable. One photon series is done basically exactly the same. It's just called one photon series. Let me see any differences here. No, I mean you're linking to an imaging plane in the exact same way. Yeah. Okay, now let's talk about adding image segmentation results. So first, I'm just going to generate some image segmentation results. So I'm going to say, okay, it's a 100 by 100 pixels, and I'm just supposing to generate this data, 20 ROIs detected. So first I'll just instantiate with zeros, which is best practice in MATLAB, and then generate random numbers for the centers of these. So this is going up to 90 to by the number of ROIs. So that gives me my X and Y coordinate. And so I'm just kind of creating a very crude ROIs here by saying, okay, well, we'll take a 10 by 10 block starting at that. I guess I shouldn't be calling it center. I should be calling it like starting point. But we'll take a 10 by 10 block and we'll say that is equal to 1 and the rest is equal to 0. That gives us kind of simulated ROIs. Okay, so we can create this plane segmentation table. Again, this is a dynamic table. Since it inherits from dynamic table, you can add optional columns. I'm not doing that here. I'm just going to do image mask, but I often see labs adding like accepted or rejected. Sometimes there are optional columns that are associated with certain processing tools like Sui2P or K-min. We're just doing a bare bones demonstration right here. So we're just doing image mask. Oh, I see a question. Can I use a single mask with, oh yeah, sorry. Yeah, we already answered that. Okay. Description, image mask, imaging plane. Yeah, again, linking to the imaging plane. Image mask. This is a kind of a new, maybe I haven't shown it yet, a different way of declaring a dynamic table. This is just doing it all at once as opposed to just creating it and then saying add row, add row, add row. You can create these vector data columns and then add the columns all at once. So here I'm just doing this all in one swoop. Okay, and then this mode allows you to provide custom descriptions to the columns. So I'm just going to call this image masks, but you can call it something more informative. Okay, so then image segmentation is a group that holds plane segmentation objects. So we'll just create this image segmentation object and then dot plane segmentation dot set. We're going to put this plane segmentation that we just created in there with the name plane segmentation in camel case. And then create an OFIS processing module with a description. Then we add the image segmentation to the OFIS module with OFIS module dot NWB data interface dot set. We'll call it again image segmentation. And then we'll add this OFIS module to the NWB file under processing, NWB file.processing.set, and then we'll call it OFIS. So now we have an OFIS processing module that contains image segmentation, that contains a plane segmentation table, that contains our imaging masks. So that's all inside the NWB file, and that will be written when we call export. Now that we have these plane segmentation results, now we can add our ROI response series. So ROI response series, as you might have guessed, inherits from time series and has just a few additional attributes. Let me see if I can find it. Yeah, ROI response series inherits from time series, points to the plane segmentation table with a dynamic table region object called ROIs. Our ROI response series also can live within a fluorescence object, which itself goes into the OFIS processing module, which is an instance of a processing module. So first, we need to create this dynamic table region, which links our ROI response series to the plane segmentation table and tells us what plane segmentation table we're referring to and which rows of that table correspond to the data of the ROI response series. So we'd use an object view here, types.untyped.object view of the plane segmentation table. And then the dynamic table region is, again, comes from types.hdmf common.dynamic table region. So here we're just saying all of the ROIs. We're referencing all of the ROIs. So when we create this ROI response series, it's going to point to every row of this table. And that's what we're doing right here. So types.core.ROI response series, it looks a whole lot like every other time series, except for it has this additional attribute, ROIs, which takes this dynamic table region that points to the plane segmentation table. All right. In addition, we're creating a container, fluorescence, which we can then, fluorescence contains our response series. So we'll go into that set object, and then we'll say set with the name ROI response series. We'll assign this ROI response series object that we just created. And then finally, we can add this fluorescence object to the OFIS module under NWB data interface, and we're setting it to the name fluorescence. And then I think this was already added, but just for good measure, we can do nwb.processing.set OFIS to this OFIS module. And then all of these data elements that have been added to the OFIS processing module, the fluorescence, which contains the ROI response series, also the image segmentation, which contains the plane segmentation. So it's the image segmentation results and also the traces that correspond are both now in the OFIS module. And then you can just write that with NWB export. You can read it back with NWB read. Again, data is read lazily directly, just as it was in the last two tutorials. So you can get this read NWB object and traverse the attributes here, get the OFIS processing module, go to the NWB data interfaces within it, get fluorescence, get the ROI response series, get the ROI response series called ROI response series. And finally, you can access data, but that won't actually read the data. It'll just tell you about the data set. If you want to actually read the data, you have to say .load, which just gives us nans because that's what we wrote here. But if you had real data, it would spit out real data. Again, if you don't want to read the entire data set, you can just index into this, and that will work. And this will read a smaller block of nans, but again, it would be real data if we had real data. If you want to access the image segmentation results, that would be under this OPHYS processing module again, inside of image segmentation, and then you use the plane segmentation attribute, and then you get the plane segmentation called plane segmentation in camel case. And then now you're in this dynamic table. You can access the image mask column of that table, and now you get all of the data. And if you want to look at the first segmentation result, then you can just do all one, which gives you this array, which you can see in the corner there, starts to do some ones. I'm using IM_show to show you that it pulls out a mask showing the ROI for that, for the region for that ROI. Yeah, and that's it for OPHYS. Any questions?